Hello, I'm Gerald Finken, and I'm CEO of E2, and I've been fortunate enough to have the opportunity to bring to you 8 webinars, today being the 8th one, on E2 Dispensing. And of course, E2 Dispensing is this new disruptive way of doing clinical supplies, packaging labeled clinical supplies. And of course, E2 is incorporating the practice of pharmacy into clinical research and allows us to do E2 dispensing. So in the past, I've had my flip charts, but today I don't have my flip charts. I'm in a different office, as you can see, so we're going to do it a little bit differently. I'm going to do the little signs, right? And so today we're going to talk about the future of clinical trials, specifically when it comes to the packaging and labeling of clinical supplies. So this is really exciting areas because it talks about the future, and that's many times where my mind lives, but we have to kind of live in reality too. And everything I'm going to talk to you isn't going to happen overnight, but it's happening pretty quickly when I think about, in particular, drug development. And they're in the hockey stick. When I talk about like preclinical drug development and what's going on with technology today, they're in the pre-hockey, no longer the pre-hockey stick or the curvature of the Moore's law. It's taking off about what's going on. And I think about the CRISPR gene and CAR T as two wonderful areas where drug development is just taking off, and how quickly those types of medications are coming to clinical trials. But if we step back and I think about my 40 years in packaging labeling, specifically with clinical trials, it's been in the last 20 years things have happened that changed and we didn't quite know what to do. So for instance, if as a project manager, if I said to you, let's talk about adaptive by design or titration studies, right? They were 20 years ago. Of course, titration studies are still ongoing and coming up. And now we're talking about decentralized clinical trials, right? Where you might need a home nurse, where you have to work with the product in the patient's home. And of course, then I want to talk about the N-of-1 type trials. So if you're a project manager and I bring these things up to you, do you start going like, oh my God, I've got a lot of work to do? Or do you say, oh, this is like an open label trial, it'll be no issue? Or what I want to share with you when you start thinking about through the webinar series is that it should always be like it's an open label trial. It should be pretty easy for you to handle these types of trials without too much complexity. So let's talk about adaptive by design studies, right? Adaptive by design. I think it was Cytel that established these types of trials back 20 years ago, and that is the patient starts in the phase 2, they go through dose escalation, right, and they have doses 5, 10, 15, 20, and then they get evaluated 2 weeks later, they fine tune to maybe the 10 and the 20, and then they fine tune to the 20, so you combine phase 2, phase 3. But of course you can also combine phase 1, phase 2, or depending on the type of adaptive by design you want to do, is how you run them. But of course, what we did was, we packaged all the supplies for all possibilities. And so I remember when Wyeth did one of the first adaptive by designs and they presented, they did 600% overage. 600%. The waste was through the roof because, of course, once a patient goes from a 10 milligram to a 20 milligram, you don't need the 10 anymore. And of course, when you think about this, that not only what you package and label, but then what do you stock at the sites for those traditional type packaging and labeling? And so, of course, I created the on-demand concept to address that, so you don't necessarily have the waste in the warehouse, but you still can have waste at the site. And of course, I'll talk a little about direct to patient so that you can eliminate that next waste. But adaptive by design, one of the flaws with this type of trial wasn't the clinical side of it. It was drug supplies couldn't meet the needs. And then the second thing was they couldn't clean up the data fast enough, so that you can move to the next level within the adaptive by design. Of course, that has changed. So let's talk about titration studies, right? Patient starts off at 5 milligram, they come in, they get evaluated, maybe they go to 10, maybe then they go to 20, and they come back down to 20, or back down to 10. And of course, that's per patient, and they go up and down. And that gets pretty complicated. So again, do you package and label all supplies? Well, usually you start doing some pretty detailed forecasting models about where that's going to go. And of course, do you stock all the sites with all the different types of product, the 5, 10, 20? And of course, then you start using your IRT to which dose do you dispense to the patient? And of course, then some of it is to use direct are used on demand again, because that again can identify which patient needs what and when. So you're not keeping things at the site in excess. And of course, direct to patient, you go right to the patient's dose that they need. But there again, we can talk about some of the costs that just becomes crazy with costs. Then we go further, and we have the new directed or the decentralized clinical trials, right? Where now we're looking at this. How do we get drug supplies to the patient? Of course, it's usually it's a decentralized clinical trials is like using on-demand direct to patient. But of course, now you have a product that needs manipulation. Where you might need some reconstitution or a mixture, where then you need the nurse there to do some. Again, I call it product manipulation work. But of course, the key there you got to remember is that a nurse, by law, licensure, is most of them only allowed to dose and administer. They're not allowed to dispense. So if they're there and the patient receives their medication, the nurse can manipulate that day's dosing, give it, but then she's got to come back, or he's got to come back the next day and do the next dose or administer. They can't stay at the patient's home with that medication and dispense a month's worth of supply to the patient. Because really that gets into the practice of pharmacy, which we'll talk about. So when I think about those methods, all right, the current methods that we're using, adapted by design, titration studies, decentralized clinical trials, and of course where we're starting to use home nurses right in the patient's home, you see pretty quickly the difficulties with this complexity. And of course the costs start going through the roof as soon as you do on-demand and direct to patient. And of course, some of the things we've talked about are wasteful. And I want to talk about time wasteful. How much time does it create to create these types of studies to ensure that you have all the supplies you need when you need them? And of course, then that goes along with very, very resource intensive with these types of trials. So moving right along, what I say is now, if you have those issues and it's complex and it's costly and it's time wasteful, what are we going to do when we start getting to the N of 1 type studies? Now, N of 1's taking on many meetings, but in essence, it's very much like the CAR-T, that every patient is their own trial. And so if you think about that, that's like you don't batch, or you have a batch record where you can produce hundreds of a drug, because guess what? It's an N of 1, so you have a batch record per patient, which is what happens in the CAR-T. The patient gets leukapheresis, they send it to the manufacturing facility, like at Novartis, and then they would activate those cells, add the medication to the, and then become CAR-T, and send them back to the patient. So then it's inject back into the patient. So it's their own cells, and so that's kind of where we start looking at the N of 1 type studies when you get into precision medicine. And now all of a sudden you have the medication itself with an N of 1, where the doctor's doing clinical assessments with every single visit, and you don't know where it's going to go. They could be using concomitant meds. They could be using different devices, different doses. It just goes all over the place. So it really becomes the one-off type of studies, similar to what the CAR-T was. Or if you think about it, it's like dose titration studies, but each patient is their own protocol. So you can say, wow, that's really going to take it and make it very difficult. So if you've listened to all my webinars, and you take a look at the way we dispense and label and work, and even my last one, I started talking about comparators, we've addressed it with E2 dispensing. It just becomes like an open label trial, because that's all you're providing is the commercial drug or the commercial device that's going to manufacture the drug, or activate it, etc. So that's really where we're going with E2 dispensing. So when I say about to start putting it into your arsenal now, so you know it, you have it, so when those studies start coming up into your programs, you can use the old methods as that'll still be going on, but the E2 dispensing is going to give you much more flexibility. And what you're going to find out is that really, when I think about these E2 dispensings, right, it's the go-to-market method. So like we do all these clinical trials, we create all this blinding, we do all these things, and then we go to market. But how's it going to be used in the market? Are they going to be doing the same methods used in clinical trials? No, it's going to be a much simplified method. And so all I'm saying is let's use that method now, which is the pharmacist gets a prescription, dispenses the medication. And if you think about it, like nurses, not a lot of people want nurses in their homes that they don't know, but they'll easily go down 5 minutes down to their local pharmacy where they know their pharmacist, and that pharmacist can do product manipulation, or have a nurse come in, or have the device, one device for 20 people in their community. Or more importantly, we have the device at our central fill pharmacy, and it gets billed there to the patient, or it gets billed there and goes to the pharmacy to the patient. All very, very simple, very, very cost effective. And that's one of the key things I say. No time waste, no money waste, no drug waste, and it's just very, very simplified. And so I hope you enjoyed the series on E2 dispensing. Well, that's my time's up. Oh, gotta go. So with all that, again, I hope you enjoyed all 8 sessions, and tune in when I start talking about other services that RxE2 is doing. Again, thank you so much for participating in these, and if you have any questions, go back to the webinars, or you can always reach out to me through LinkedIn or through my email address at gfinken@rxe2.com. Thank you very much. It's been a wonderful time presenting E2 dispensing to you. Thank you.